Hello, everybody. This is the recording of the first lecture on blood vessels. This is chapter 21. This is titled the hemodynamic chapter, and that tells you that blood vessels have a lot more to them than just being confining pipelines for blood flow. So we're going to look at properties of small and large blood vessels, and we'll be looking at a particular surprising design of capillaries, and that design is to be leaky. And we're looking at the consequence of that leakage as the first major physiological role for these blood vessels. So let me do a screen share. And in the screen share, let me pick up a laser pointer here. So, yeah, maybe we're going to make this full screen. Right. So, blood vessels are seen on models, and we'll be seeing this in the lab, both for arteries and veins. The blood vessels are highly easily visualized as pipelines for blood flow. It will sound very familiar to you that we have arteries and veins. If for nothing else, from the heart lab, you have seen coronary arteries, coronary veins. On that heart model, you have seen pulmonary veins, you have seen pulmonary arteries. So before we go into structural aspects of these blood vessels, it's really good to identify them by their anatomical definition. So arteries, and this is really important distinction, as opposed to color coding, arteries are always carrying blood away from the heart. So the flip side of that, veins are always carry blood towards the heart, back to the heart. These are the two anatomical definitions. And once you look at branching of the arteries, if you go to a microscopic level, you're going to see smaller versions of arteries, arterioles. And then eventually arterioles will branch into many, many capillaries. We'll see capillaries as the smallest blood vessels, and they have the simplest structure on the outside. And a very similar thing happens to veins. When veins carry, collect blood from tissues, initially they start out as venules. These are tiny or small diameter version of veins, but eventually all the venous blood flow will be collecting into large veins. You have seen these on the heart model, if nothing else. The bottom line here, vasovasorum, is a curiosity. Sometimes the veins and the thick arteries have so thick walls that the walls of these blood vessels need separate oxygen supply. And so these vasovasorum are small, tiny blood vessels that supply oxygen and nutrients to the thick walls of big arteries and big veins. We'll be looking at structural differentiation of arteries and veins, and the structures that we see making up the blood vessel walls must be fulfilling the functional requirements. And for arteries, we want them to be elastic. Think about taking a pulse. When you take a pulse, there is a little ballooning of that artery. A lot of the times we take pulse on the radial artery. So let's take that as an example. When you feel that hit of the pulse on the radial artery, It comes from the temporary ballooning of that radial artery to push on your Meissner corpuscle. But after that hit of the pulse, we expect the artery, the radial artery, to go back to its original shape. And that is possible because of elasticity. Some structures in the walls of arteries will bring this property with them. Another expectation of arteries is contractility. So increase or decrease the lumen of the artery, we'll be calling the increase in the lumen size vasodilation, decrease vasoconstriction. And you have seen a role for decrease in blood clotting, hemostasis, that vascular spasm was nothing else but the expression of contractility. Smooth muscle cells narrow the lumen in case that blood vessel is cut. So, and we'll be seeing roles for vasodilation, vasoconstriction later chapters as well. So these two properties, elasticity and contractility, are coming from the walls of the arteries. When we look at the actual layers, if you love the tunics of the eye, you'll love the layers of the arteries, and also veins have the same layers, but let's focus on arteries here. Here is the lumen of the artery, and this lumen is lined with this simple squamous epithelial layer, the endothelium. This is the endothelium that needs to be covering the area very tightly. Any injury to this endothelium will start blood clotting, platelet plaque formation. You can see nice evenly paced cells here in the endothelium. Next to, just outside of the endothelium, there's this internal elastic lamina. This is one of the layers that provide elasticity to arteries. These two layers, endothelium and internal elastic lamina, form the tunica interna. When we go a little bit further outside in this wall of the artery, we find a thick layer of smooth muscles. This is at the beginning of tunica media. Right outside of the smooth muscle layer, there is the external elastic lamina. And when we go further outside, we arrive at the tunica externa. Tunica externa is just connective tissue separating blood vessels from their environment. There are some arteries, especially very large ones, that are called elastic arteries. On that heart model, any parts of the aorta, whether ascending, descending aorta, aortic arch, the abdominal aorta, are all examples of elastic arteries. This just means that elastic elements are very thick in the wall of these arteries. These arteries expand hugely after every heartbeat, because into the aortic arch, 70 milliliters of blood is shoved into every 0.8 seconds with every heartbeat. So this aortic arch needs to expand and then push this volume of blood further down the aorta. So regaining the shape of the aortic arch is the consequence of thick elastic layers in there. So aortic arch and the other parts of the aorta qualifies as elastic arteries. And then there are muscular arteries. Muscular arteries have a very thick, smooth muscle layer in the tunica media. These are smaller arteries. We'll see quite a few of those in the lab when we name hundreds and hundreds of arteries. And we'll be naming gastric, superior mesenteric renal, pulmonary arteries. These are all muscular arteries. These arteries can exceptionally well vasoconstrict or vasodilate, and they distribute blood flow. These muscular arteries can decrease blood flow to organs of the digestive system during fight or flight response, for example. Gastric, superior mesenteric, inferior mesenteric, splenic arteries contract, and that vasoconstriction slows blood flow to those organs. When we follow these arteries and follow them to their microscopic size branching, we will be coming onto the arterioles. So the tunica media thins out, still has some smooth muscles, as you see, these strap-like structures. So arterioles can still vasoconstrict or vasodilate very efficiently. And for a particular organ or for a particular part of an organ, arterioles can direct blood flow to through their dilation. Let's say a few words about veins. Veins have the same three tunics or tunica layers in their walls, except that the tunica media has a thinner, smooth muscle layer. In the lab, we will see an artery and vein coming from a tissue right next to each other, and it will be striking how much thinner the tunica media is in veins. The veins also don't have external or internal elastic lamina. They have elastic proteins scattered there, but they don't form a visually identifiable layer. A unique aspect of some veins is that they have valves. You can see two valves here. Valves, just like inside the heart, valves inside the vein prevent the backflow of blood. This is especially useful for veins of the limbs. Imagine for a minute that right here is our ankle, and right up here is our knee area. So this vein represents veins that are going through our leg. Gravity would pull the blood all the way down to the ankle, but we cannot have blood stay put in our circulation. So we have to somehow make sure that blood is moving upward. And this is going to happen by the contraction of the leg muscles. When this middle section of this vein is under pressure, imagine there's a muscle to the right, muscle to the left. When they contract, they squeeze on this vein, where the content of the vein, that the oxygenated blood cannot go downward because the valve is closed, but it can move upward. Pressurized blood here would open up this valve, and pressurized blood would move against gravity. So these valves are very instrumental in moving blood away from the lower limbs and maintaining active venous circulation from those lower limbs to structures further up. For the venules, we see a thinning out of the blood vessel wall. Again, just like in arterioles, the tunica media contains much fewer smooth muscles. Venules are the place where neutrophils and monocytes can exit the blood vessels, and going through this endothelium is quite a feat when these white blood cells exit. In the immune system chapter, we'll see a good illustration of how that happens. When white blood cells want to exit blood vessels, these venules are a favorite spot for them. So here is a little fact about veins and arteries. When you measure blood volume, as you're sitting and listening to this lecture, Most of the blood, almost 70%, is in your systemic veins and venules. Blood can be pushed out from these veins and venules when we need it. Exercise, muscular activity, fight or flight response, and blood will be moved eventually into the main circulation. We talked about regulation of stroke volume way back in the heart chapter. So blood diverted from sedentary veins can come to the right side of the heart. And when the right ventricle is stretched more by these extra blood that's coming from the venules, that right ventricle can respond using the Frank-Starling law and increase stroke volume to handle this kind of situation. So the systemic veins and venules are called blood reservoirs because of the ability to confine large volumes of blood. The walls of the veins are thinner and therefore more stretchable, so blood can stay in them and circulate slowly. Let's take a look at the tiniest blood vessels, the capillaries. So the capillaries are a link between arterioles and venules. We have thousands of miles of capillaries in a body. So is this the next slide? I just want to go back here for a minute. Here's an electron microscopic view of the capillary bed. And you can see the larger arterioles here, but the smaller branches are forming this jungle of capillaries. The diameter of the capillary is just a little bigger than a red blood cell. So that kind of gives you an idea how skinny these vessels are. Think about that blood smear slide that you saw in the lab. You've seen hundreds of red blood cells on there. Imagine blood vessels as thin as those tiny red blood cell donuts were on that blood smear slide. So these vessels are incredibly small in diameter. Capillaries come in different shapes, and we also can have different densities in different places where there's a lot of oxygen, excuse me, when there's a lot of oxygen and nutrient flow that needs to come out of these capillaries. We have a large density, very dense arrangement of these capillaries in skeletal and cardiac muscle, liver, kidneys, brain. We have a very extensive branching of these capillaries. Now, I want to point out something. We'll come back to this in a few minutes, but on this drawing, something is missing. Take a look at these large wide gaps. This is where dozens of cells reside, and all those cells are waiting for their oxygen and nutrient to be delivered to them. So in tissues where the cells are very active, like in skeletal muscle, the cells contract. In the heart, the cells contract. In the liver, the cells are carrying out chemical reaction. So all those activities require oxygen and nutrient supply. And so the capillary density is very high in those tissues. So oxygen and nutrient can be brought to these very active cells. Now, on the flip side of that, there are places in the body where there are no capillaries. In epithelial tissue in general, our cornea and lens of the eye is largely free of capillaries, and also cartilages have no capillaries. What is the function of capillaries? In addition to, excuse me, in addition to bringing all the oxygen, nutrient, blood to these clumps of cells, Capillaries are the ones that allow oxygen and nutrient to come out of the capillary and move in between these cells until the oxygen and nutrients are removed by the dozens of cells in these gaps. We'll see how that happens. This process called capillary exchange is this exchange of nutrients and wastes. We'll talk about that in a few minutes. Capillary walls, and you'll see this here. Capillary walls are extremely simply constructed endothelial cells, as you see here, and a basement membrane. That's all you need to make a capillary. And to look at the size of a capillary, here are red blood cells. It's just barely fitting through this capillary. So capillaries are real tiny, the tiniest blood vessels in the body. Many places in our body have these regular so-called continuous capillaries. Take a look at the border between the cells. This is kind of straight, and when fluid is oozing out of this capillary, and we'll see that these capillaries are designed to be leaky, fluid can go in between the cells. So oxygen, nutrients are all leaving these blood vessels by fluid flow coming through these gaps. If we have a need for larger fluid flow between the inside and outside of the capillary, here is another design. Fenestrated capillaries just mean that there are small window-like structures in the body of the capillary, the simple squamous epithelial cells, and through these openings, fluid flow accelerates. Now, why would we need fenestrated capillaries? Let's take a look at a few places where we have looked at already. Think about chapter 18, endocrine glands. Imagine that this blood vessel is going through a pancreatic islet. If the pancreas wants to secrete insulin into the bloodstream, insulin molecules need to find a very efficient way to get into the blood. So from an endocrine gland, fenestrated capillaries always assist of hormones entering into the bloodstream. Let's take a look at ciliary process. We looked at this in the eye, where ciliary process is making aqueous humor. So if this is a capillary inside the ciliary process, aqueous humor would be coming out of this capillary, and aqueous humor is nothing else but filtered plasma. Whatever comes out of these fenestrations of the capillaries, that is what we call aqueous humor from the ciliary process. We'll have some other examples here in the digestive system. We'll come back to small intestines. In the small intestine, nutrients, electrolytes, water needs to be absorbed from the small intestine into the bloodstream. So in the capillaries of the small intestine, the fenestrations are helping the movement of glucose, amino acids, fatty acids, vitamins, electrolytes, water from the lumen of the small intestine into the bloodstream. And the last, the weirdest form of capillaries are called sinusoids. We'll be seeing one set under the microscope after our second practical in the case of the liver. But in addition to the liver, other organs also have sinusoid type capillaries. The fenestrations, the openings are huge. So let's take a look at a few examples where sinusoid design come very handy. So let's go back to the liver and spleen. You have seen this in the blood chapter, that in the liver and spleen, from the circulation, worn out red blood cells need to be taken out. So those macrophages that do the recycling, they are sitting in these gaps, in these fenestrations, and reach into the blood vessel and grab malfunctioning red blood cells for recycling. In the case of bone marrow, let's mention this as the flip side. Now you know from, yet again, from a blood chapter that our bone marrow, red bone marrow, is making 2 million new red blood cells every second, and they shove it into the circulation. How can you shove red blood cells into the circulation? Well, the answer is sinusoid. So imagine that the red bone marrow tissue is sitting out here and hemopoiesis is going on outside. But once those red blood cells are ready to be used, they are pushed into the sinusoid through these openings. And this is how we get new red blood cells into the circulation from a red bone marrow. So each of these types of capillaries have or can have a particular role in accelerating movement into or out of the capillary. And this movement in and out of the capillaries come to the fore with this physiological event of capillary exchange. So this is movement of materials in and out of the capillary to a very large volume amount per day, while the cells are taking up oxygen, glucose, amino acids from interstitial fluid. The oxygen, glucose, amino acids can be brought to those cells very, very efficiently by bulk flow. So let's take a look at bulk flow, and this is best illustrated on this image. Now you see the gaps of the capillary bed filled with cells. This is a really good look at how our cells are arranged inside our body. And these brown cells could be neurons in the brain. These could be cardiac muscles in the heart. These could be skeletal muscles in any kind of skeletal muscle. These could be liver cells, hepatocytes in our liver. So take a look. Here comes oxygen and nutrient-rich blood from the arteriole. And an event called filtration will take place where, to the tune of 5 gallons per day, take a look at this arrow, fluids will be squeezed through the wall of the capillary, and fluids from the blood will be entering the capillary bed. This fluid coming out of the capillary is rich in oxygen, is rich in nutrients, and so the cells that receive this flow are going to be well fed. Their oxygen demand is satisfied. So this kind of flow, here's another arrow showing the same direction, movement, representing the process of filtration. We have the filtration of about 5 gallons of tissue fluid into living tissues every day. What happens to the accumulated fluid here? Well, there is a flow in the opposite direction. Take a look at this arrow, or this arrow, or this arrow. They represent a process called reabsorption. Reabsorption is a movement of tissue fluid back into the blood vessels. And reabsorption is bringing back about 4 gallons of fluid per day into the venules and the beginning of the venous circulation. So 5 gallons of fluids are filtered, 4 gallons of fluids per day are reabsorbed. This leaves us with 1 gallon of fluid staying among the cells. Now that fluid could accumulate to dangerous levels, so it is absorbed into the sort of lymphatic structures, lymphatic vessels, and so that accounts for the drainage of the leftover, or the 1 gallon difference between the filtration and reabsorption. So capillary exchange is nothing else but exchange of materials. Here is one part of the exchange, the filtration, and here's the other part, reabsorption. Filtration and reabsorption creates movement of large volumes, and large volumes of fluid can carry a lot of oxygen, a lot of nutrients in between the cells. All our organs are dependent on filtration to receive the proper oxygen and nutrient. So here we are looking at the driving forces. So take a look at the arterial end of a capillary. BHP stands for blood hydrostatic pressure or blood pressure. It's about 35 millimeters of mercury here. BCOP stands for blood colloid osmotic pressure. This is a force that invites fluid flow back into the blood vessel. When you look at the arterial end of this capillary bed, the blood hydrostatic pressure is larger than blood colloid osmotic pressure. And so this 10 millimeter of net outward pressure is going to drive filtration. So net filtration pressure, take a look at this blue arrow, the sum of all the other forces is pointing out of the blood vessel. So fluid stream would be moving out of the arterial end of the capillaries to a tune of 5 gallons per day. How are we going to get that 4 gallons of fluid back into the venules? And on the venous end of the capillary, blood hydrostatic pressure drops. Blood colloid osmotic pressure stays the same as before. So now the balance of these two forces is tilted towards the blood colloid osmotic pressure. And this means that fluids with electrolytes, with small proteins, with waste products will be streaming back into the venules and the venous end of the capillary bed. This is reabsorption. And so again, the pressures promoting reabsorption is turning out to be the dominating blood colloid osmotic pressure. Now, what is this pressure? What is blood colloid osmotic pressure? It's the attractive force of proteins that stayed in the blood vessel because of the high concentration of protein that is attractive for water molecule movement, and water and electrolytes and other molecules will move from the outside of the venous end of the capillary into the venous end of the capillary, and therefore fluid will be reabsorbed to the tune of 4 gallons per day. So keep in mind the driving forces at the arterial and the venous end of the capillary bed. Different forces result in movement of fluid in or out of the blood vessels. And as a last part of this lecture, let's mention the term edema. You also are becoming world expert with regards to edema. We have mentioned this with the aldosterone hormone. We have mentioned edema with congestive heart failure. So edema is the abnormally large accumulation of interstitial fluid. It usually means swollen body parts. So when edema comes from the malfunction of the capillary bed, mostly it means excess filtration. Somehow the driving blood pressure is so high, mostly indicating some level of hypertension. And so fluids are moving into the capillary bed at high speed, at much higher volume than much higher than 5 gallons per day. And accumulation of the filtered plasma will show up visually as edema. We also can cause edema by slowing down reabsorption, either a defect in plasma proteins or some kind of a obstruction of lymphatic capillaries can cause edema on the reabsorption end. So this was a major physiological aspect of capillaries, to carry out capillary exchange and allowing those two forces, blood hydrostatic pressure, squeezing plasma fluids out of the capillary, and on the venous end of the capillary bed, allowing blood colloid osmotic pressure to attract water back into the venules. And this is a place where venous circulation starts from. So this was the important contribution of blood vessels in the capillary bed, allowing capillary exchange to happen. The next topic here will be, next lecture will be all related to blood pressure, and we'll leave that for the next lecture. So I'm going to stop sharing here. And next week's lecture will be completing this chapter 21. If you have any questions, please don't hesitate to message me. I will ask you in our next lecture, how did you like the introduction to blood vessel function? And then we'll move on to discussion of maintaining blood pressure. So I'm going to stop recording, and I'll see you guys next week.